afternoon to all. So I think no today 12 totally how many people are there in a batch, right? 15, right?
Yes, sir.
15, sir.
Yes, sir.
15. So I think only 12 members, that is including me. So it is excluding it is 11 members joined. Shall we wait for 2 minutes? Let the other joins, right?
Yes, sir.
Yes, sir. Okay, sir.
Okay, fine. So we'll wait for 2 minutes. Okay, let everyone join. Then let me start the class. Thank you. Hmm. So I think 14 members join. Okay. So anyway, no, good afternoon to all. So today we are going to discuss about the part of CAD. I think no, you might have already learned what is CAD, what is the application of CAD, all those things. Okay. Now what we are going to do is it audible?
Yes, sir. Audible,
Yes,
sir.
sir.
Okay,
Yes, sir.
okay, okay,
Yes,
okay,
sir.
okay, okay. Thank you. So what we are going to learn today in the sense, please try to understand. Today we are going to actually this is the first chapter of CAD, okay, or first basic content of CAD. We should know how the computer is applied. I think you know about CAD, right? Can you able to give me a few points about CAD or what is the application of CAD or something like a few points you learned in CAD? Anyone, please give 1 or 2 points. That is sufficient. Just to have an introduction about the class. Any 1 or 2 points about CAD. Eh, Any 1 point about CAD?
The design of experiments, like
Excellent. Good. Sufficient. Okay. Thank you. So that's what no like in CAD we are focusing more focused about design of experiments and then no doing experiments with the support of computers and then no reducing the human burden or pressure on human. Okay, this is what actually the CAD is developed. Okay, and then we can able to reduce the time and then resource and then expenditure of money. All those things we can able to balance by using this application, computer application. So before entering into that, we should know how this idea created, how this happened actually, how the computer started entering into our domain, pharmaceutical domain, and then how now it became one of the unavoidable resource in the pharmaceutical domain. Computer and software, no, now it became like unavoidable resource in pharmaceutical domain. How it happened and then how this went, all those things, no, we will be today we will be learning. Okay, more or less it is going to be like a history class or it is going to be like no science driven history class. So try to adjust. If you feel bored, you can tell me or you can ask me so that no we will try to make the class more interesting. Okay, so now I'll share my screen. Okay, I think you can see my screen.
Yes, sir.
yes, sir.
Okay.
Yes, sir.
Yes, sir.
So yeah,
Yes, sir.
oh good. Okay, so computers in pharmaceutical research. Computers in pharmaceutical research. So about that we are going to discuss. First, we will be discussing about the introduction, and then we will discuss about the history and present scenario, how this idea started, who initiated this idea, and then now where we are in this computer and pharmaceutical development, and then use of computers in pharmaceutical R&D. So I'll tell you like on what are the areas specifically related to pharmaceutical areas. Okay, pharmaceutical history is more or less related to chemistry, and then when it comes to use of computers, we are going to exclusively discuss about how this computer applications are used in pharmaceutical domain. Okay, and then you can also imagine like where this field is going, and then what are the new job opportunities you may be getting because of learning these softwares. Okay, all those things, and then the ethics of computing. I think it is already covered. Now, next we will discuss about how to use internet properly, or on what basis this surface engine and then search mechanisms happens to get in information. For example, if you want to do a research, first of all, immediately despite of going library, we will start in initial stage, we will start searching with our computer or an internet. Okay, so how this working, what is the backend mechanism happening in information retrieval systems. Okay, mean search engines, all those things, and then how we need to give information so that we can get the best outputs. About that we will be discussing, and then like a pattern search and then plagiarism deduction, all those things also we will be searching, like we will be learning like how to check the plagiarism and then how to get information about the patent. And then for example, you may be having one innovation idea, innovative idea. Okay, you might have some idea, or you might have some experimental model, something like that, which you feel it is innovative. So that you need to check it, right? Then how to do the search, and then how you will come to a conclusion that this product is patentable. Okay, about that, and then computer and market analysis, the last part, how it is used in market forecasting, market analysis, all these things. Okay, this is, don't think this is not related to pharmaceutics. This is more or less, it is very closely related to pharmaceutics. Once we enter into the chapters, you will get an idea. So about all those things we will be discussing today. Okay, so let me go to the chapter. I think you might have got book. Okay. So first, first initial stage, what happened in the sense, initially we may think, no, the computer will be computer. Initially, it is how the pharma sector used in the sense, we used pharma sector used computer, but not in term of research. We started around 1940s. No, 1940s, the computer started entering. It is like, more or less, it is like a story. Please try to understand. Around 1940s, people started thinking about a computer in all fields. Most of the fields, they started utilizing the computers. And the same, no, in that same way, the computer started entering into the pharma domain also. But surprisingly, these things, no, initial stage, they have not entered for scientific purpose. Those computers were entered into pharma sectors for non-scientific applications, like for example accountancy, and then, no, like calculations, these, like in calculator, like in calculator in entered. And then late stage, yeah, outbreak happened after 1940s. No, there were one big outbreak. What outbreak happened in the research in the sense, people started thinking molecule and then receptors are related. Okay, I think you are very well about mechanism of action of drugs, right? A particular molecular structure have an ability to bind with specific receptor, and then it may trigger a receptor or it may suppress the receptor. Okay, like then it will produce some therapeutic action. So this mechanism or this process they established, and then they found in elaborate way in 1940s and later late 1940s, and then they came to know, okay, fine, it is again everything based on chemistry, the total body and then drug mechanism and how the drug works in the body, all those things based on chemistry. This clue research is found. Okay, then automatically once you can able to quantify the chemistry, automatically we can able to predict by using mathematical models. Okay, please try to understand what I'm intending to tell in the sense, computers initially they used for accountancy purposes. Okay, after that researchers found the relationship between molecule and then receptor, like lock and key mechanism. This particular molecule binds to a particular receptor and then it can produce some therapeutic action. Okay, then they found the structure of molecule, they found structure of receptors also. So everything they found. So now it is time for us to incorporate this information into a computer, and so the computer can simulate and the computer can predict what is going to happen. So this way people started thinking only after the discovery of molecular structures and then drug receptor structures, how it binds and then how it produces action. This is the stage one, how the people started thinking the computer can also be used in predicting biological activity. Is this clear till this?
Yes, sir.
Yes, sir.
Oh, okay. Now we'll see. Now we'll see. Now from how it happens on from 19s. Okay, so 1940, this basic idea. After that, people started vigorously working on this hypothesis, and then, they got it was enormous information. And then in 1940s, mid of 1940s, say 90s, okay, mid of 19s, around 1950 or 1955, in that basis, no, people started working on QSAR model. Okay, like structural active relationship. I think you might have learned in your chemistry, structural acting relationship in the sense very simple. You take a paracetamol molecule, you remove one group and then attach one more functional group, and then you can check whether it is binding to the receptor or not. If it is binding more powerfully to the receptor, then we can predict it will produce more or better action than the our old molecule. So this new molecule you can push into the market. Okay, this is known as structural acting relationship. Like, the structure of molecule, and then the structure of receptor, how it will bind. On changing the structure, how it can bind, all those things, we can able to predict. So after 1940s, 19 mid of around 19s, mid 1950s, 1955s, people started thinking of that. And then still now I'm telling you, typically in those phases, computers have never ever entered into pharmaceutical domain. Okay, computers were only used on simulating the molecular docking, how one molecule will attach with the another molecule, then how one molecule will attach with the receptor, those things. this reduced enormous workload of a chemist. I'll at the end of the history, I will tell you how initial those days, how they will do the research. After that, you will understand how the computer made a very big impact on reducing the work burden of those chemists. Okay, so now you please try to understand the major outbreak happened is QSAR research mid of 19th century. Okay, now 1960s. 1960s, what happens? No, companies started working into these areas. Companies started working into this area. The first company who initiated the research is, I think, Eli Lilly. Eli Lilly is the first industry who initiated the research. And then once what happens? No, Eli Lilly, what they thought, they did enormous investment, and then they did research on this molecular docking, all those things. Unfortunately, Eli Lilly failed. Eli Lilly failed in the sense, I think, what is the first molecule they planned to develop by QSA or by predicting docking? First, anyone knows or anyone heard of that? What is the first structure or first molecule?
No, sir.
Hmm? Okay. Huh? Yeah, exactly. Beta lactam antibodies. Okay. So beta lactam. This actually, now you think that beta lactam credits should really go to the Eli Lilly. But what happens? No, Eli Lilly dropped the experiment. They thought they were spending a lot of money without any productivity, because those times how it ran. What happens in the sense, actually, synthetic chemistry as a competitor, they were doing traditional method of molecule development. On one side, this Eli Lilly were developing a tool for developing the molecules. Okay, please try to understand. One side, a synthetic chemistry group of synthetic chemists, scientists, they were using traditional method for molecular development. And then one more side, our this quantitative or computational chemists, they were developing in their own style. Okay, the quantitative or computational chemists, being a new area, they took little bit more time. So the synthetic chemist people were fast, because it is already practiced by them, right? So they did a fast outcome. What happens then? Finally, even though the company did a big achievement, but they spent a lot of money, and then they thought this will not work out, and then they dropped the project. They were forced to drop the project. Clear? And then, like, other companies, they also understood, and then they started thinking of that. So this gave a very big awareness. So now then around 1960s, it became a very kind of, how I can tell it, became like a um, oscillation in the pharma sector, whether do the computer will really help a mankind in pharmaceutical development, or the computer was a myth. Okay, in that case, so the people started thinking. And then now what happens, the big shot computer and then software manufacturing companies started coming, like IBM. Okay, IBM, these kind of big companies started supporting these kind of projects. Okay, and these things initiated in 1960s. Okay, IBM, all those big shots, they started in 1970. What happens as an outcome? They have developed protein data bank. It is, you can check even now, the protein data bank. And then Cambridge structural database is not, it is available, but it is not used that much. But this protein data bank introduced in 1970. I think, have anyone searched this protein data bank? What is the use? No, no, if you know, no problem, you can tell. If not, I will tell you. It's not a big deal. Just know, it is just to know whether you know or not. Nothing else. Okay, well then, good. So protein data bank, in the sense, in biological system, we have n number of proteins. Certain protein will be a receptor, certain protein will be a transcellular receptor, will be an intracellular receptor, and then some proteins will be blood proteins. Like biological system, we have enormous number of proteins. Even we will tell receptor as a protein. It contains a series of amino acids, right? So what happens in the sense, these big shots started entering into this computer as an pharmaceutical application. Once after they entered, now what other researchers started doing, everyone started simulating or developing graphical proteins. Okay, what they will do, they will check what is the human protein structure. Accordingly, they will develop a graphical, right, guys, like 3D models, and then 2D models, ChemDraw, like this number of softwares are there. All these things were developed by big companies. With that support of the softwares, these people, what they will do is they will develop a 3D model of protein. They will develop, and then they will keep in the protein data bank. Even today, you can search in protein data bank. You can able to, if you can, I think some protein name. You may be knowing the protein name of albumin. You may be knowing PGP. Like this kind of few ITM, I don't know how far. Okay, few protein names, right? All this protein name you can search in PDP, protein data bank. You can able to find how it looks. Okay, 3D homologous model you can able to see. In that way, proteins, they start those things in 1917s, they started, because on that time, even though this part failure, but still computers were used for QSR, structural activity relationship, the computers were used, mean which structure of molecule can bind with the which type of protein. That kind of science studies they were continuously doing to facilitate that study. To facilitate the study, number of protein structures were being unwrapped, discovered, and then they kept in those protein data bank models. Okay, these things know that even now it is highly useful for research community. Once you started seriously entering into a deep research or some realistic research, you will understand this protein bank is like kind of no god for us who supports in all research aspects. Okay, such a home, all those models they started putting into this protein data bank. But what is the actual problem? Those days pieces were not developed. Pieces were not developed. They were using DOS, disk operating system. I think you know about OS. Now we are using, most case we were using like Windows. These OS we will be using, but those days this kind of user friendly Windows were not developed. Okay, that graphical and the user friendly Windows were not developed. Those days most of the people were using DOS. Okay, that is quite difficult to handle. Even now, the protein data bank is well and good, started settling, contains a lot of information. But apart from all those positive things, the computers, what people were using were DOS, and then it is not user friendly. It is quite difficult for them to correlate the study and then get the result. This problem they were facing. Then automatically Apple entered into the game, because Apple were very good in developing user-friendly computers on those days when comparing to DOS, advanced and user-friendly things. And then Apple came, so Apple also supported our technology. And then at the end of the day, now the pharma company started getting a user-friendly, good QSAR, protein modeling, and then drug binding, dynamic undocking. All those studies can be done in such a way. No, slowly, steadily, the growth started coming in 1970s. In global pharma sectors, got a clue, started understanding how to use this particular computer towards drug development. Okay, now after 1975 to 85, this started vigorously. This started vigorously in the sense, like very simple. I think you can think in these times. In these times, most of the companies started funding, and then like most of the organizations started funding, taking employees who was those time it is like a golden period, because companies started understand by using computer I can reduce the effort. Once if I optimize the program with that process, first optimization is quite difficult, but once optimizing the program, with that program, I can run hundreds and thousands of molecules. It will reduce a lot of burden. So they started recruiting. So they started recruiting number of people who do have very good idea on computer programming, who do have good idea on molecule structures, chemist, and then computational idea. In this compo, they started recruiting a number of people. So now the people started coming to this particular area and then started working on this. And then 1990s, the actual game begins. 1990s in the sense, this is the time of supercomputers. Time of super, Cray Research Company. I think there are a few companies who started working on supercomputers. They developed a supercomputer, but they don't know how to commercialize it. Do anyone knows what is the use of supercomputers? You can give your own hypothesis. I'm not expecting any answer. Just to tell any answers, I'm ready to accept. Do anyone know what is the use of supercomputers? Why they're using? Have you heard the term supercomputer? Have you ever heard the term supercomputer term? Supercomputers.
Yes, sir.
Excellent. Okay, except those Hollywood and movies, something like that. You tell me what is the application of supercomputers? I'm see. Listen, I will never ever expect answers. I want you to involve in the class. That's all. You please tell me what is the use of supercomputers,
So
according to you.
the personal computers we use are they work at a very limited speed, sir. We can maximum share information on GB basis, like GB per second. But supercomputers, they are used to gather information like in speeds of GB per seconds to store more information that like 10,000 personal computers can store, sir.
Excellent. So you are telling what is the difference between our regular computer and supercomputer. My question: what are the areas and what are the sectors these things are generally used?
Sir,
The performance will be good, sir.
Excellent, Ganna. So really, I'm accepting good. Okay, supercomputer, the speed will be good, then performance will be good, and then the like you can run n number of programs. Likewise, no, the frames, everything will be more faster than regular computer. I'm accepting everything. But my question, where these things are actually used? See,
Sir,
a
cloud
regular.
servers,
Yes,
cloud
please,
servers
please,
like
please, please.
Google
Exactly,
Drive, those and all they
exactly.
use supercomputers, sir.
Yes, okay. So yeah, cloud servers and then no like head offices of those server maintenance and the information retrieval system maintaining head offices, aerospace, and then like these areas they were using. Clear. But unfortunately, 1990s these areas were not so developed. They are in developing stage. So now the companies, they started developing supercomputer, but they don't know where to fit this particular computer, and they want to increase the sales. See, you think about the IT companies, no, they were investing a lot and then they have developed computer. Now they want to find a way where they want to sell the computer. And then they came to a concept of tying up with the pharmaceutical company, and then they want to make a supercomputer as a super drug discovery tool. Okay, this happens in 1990s. In 1990s, what happens in this sense? Again, we were discussing about the key player. Lilly was the key player who initiated this project, but it was failure. So now they tied up with Eli Lilly, and then with the support of Eli Lilly, they were using their software. They're using their supercomputer, and then they are making it as a some super drug discovery tool. Okay, now you see they have protein data bank, we have protein data bank, we have a softwares, we have very good machine who is ready to run n number of operations, or we is ready to take any big operations in a very easy way. Likewise, now the complete combo was set and then it was ready. Automatically what happens in 1990, it was a very big kickstart for computer application in pharmaceutical sector. Once after this kickstart happened, many companies like Bristol Myers, I think you know about Bristol Myers, it is one of the world's biggest company. No one knows the name, but this Bristol Myers is, I will tell I used to tell Bristol Myers Squibb is father of all pharma companies. Their only role, they will do research, they will develop a molecule, and then they will trial the molecule, and then they will go to big companies like GSK, Pfizer. These people will sell it. Okay, so I'll tell just an example. Okay, in layman terminology, Bristol Myers is a kind of a father of all companies, something like that. So even these kind of Bristol Myers, Johnson Johnson, Merck, Bayer, these kind of core research-based driven companies started using this technology for fastening their research. Okay, so till 1990s, these only complete computers are maximum they're used in understanding or analyzing how the molecular way the chemistry happens, and then no molecular support and drug discovery. Still the formulation touch not it came. Okay, after that around 2000s, okay, late 1990s 2000s, they started thinking of computers in formulations development, all those things. Okay, those things happened recently in current scenario, but you try to understand till 1990s people are more focused on computer as a aid for drug discovery. Okay, so now we'll see apart from all those things in R&D. Now we'll see pharmaceutical R&D. Okay, how the computers are used. First major important utility of computer is information management system. Information or data analysis is a data management tool. It is used as data analysis and data management tool. Why we have given this is first in the sense, this is what foremost important thing in pharmaceutical sector. You please try to understand why the pharmaceutical sector needed highly educated people in the sense, because the only major important in the sense, the good manufacturing practice, good documentation practice. A good documentation practice will support the good manufacturing practice. How we can tell it is it's GMP in the sense, once you have a good documentation, once you have a good follow-up, and then once you have a good retrieval system, then we will tell it as a good manufacturing practice. To get good manufacturing practice, the industry should have good documentation practice. To have good documentation practice, we should have a good tools to maintain these documents. Okay. Are you getting what I'm telling? GMP is very, very important for all pharmaceutical sector. Okay, even for research and then for even manufacturing, it is very, very important. To have a GMP, good documentation practice is important. GDP. Okay, to have a GDP, we need a good computational data management tool, and then because manual data management is okay at some part, it is acceptable. But when you started producing in bulk, and then when you started doing turnover of 100 crores, 1,000 crores, 2,000 crores, you need some good data management tool to avoid errors and to track the work. Okay, so those cases, these computers are nowadays used in a very, very elaborate way. Okay, so there are three major softwares: chromatography data system, and then laboratory information management system, and then like a text information management system. These three are the major software in data management tool. I mean, I'm talking about R&D. Please don't think, even despite of that, in pharma companies, accountancy, and then like HR, human resource management, all everywhere computers are using. But now we are specifically focused, then we are studying on pharmaceutical R&D, research and production based area. What are the things are used? Okay, so one most case we will be using these three system, like chromatography data system, laboratory information management system, and then text information management system. These are the three things we in most of the pharma companies they'll be using. Clear? So this can be used, data are used for regulatory filing, even no regulatory filing, and then audits, all those things they were using. Okay, now we'll talk about chromatographic data system. Chromatographic data system, in since I think you know very well, 99.9% all pharma company will have chromatographic devices. Okay, that can either be a gas chromatography if it is a volatile substance, or they may have a high performance HPLs, high performance liquid chromatography. Okay, so or else they may have like gel electrophoresis, supercritical chromatography. Based on their requirement, the pharma companies and then pharma research lab will have their chromatographic device. I think it is 99.9 all pharma company who are doing pharmaceutical research may have these device to support their research. Once if they are having this device, then chromatographic data system is very, very important to understand what is happening, and then to store the data in a perfect order, periodical way, easy to retrieval. All those applications, these softwares will be used. Okay, when they go for any regulatory filing, they, because those nowadays the softwares is like logged softwares. We cannot able to duplicate the data, or we cannot able to manipulate the data, something like that. No, it will be systematically perfectly saved. Okay, even now like USFDA, they were asking these kind of storaging technologies automatically. I mean, automatic come closed data system. It is like you cannot able to change the date, time, or manipulate. All these kinds will never happen. Clear? So most of the companies, those who have research laboratory, they will have these kind of data system to maintain their analytical informations. Point one. Okay, next, laboratory information management system is again one more critical management system which is available in pharmaceutical sectors. Clear? So this here in a pharmaceutical sector, we don't think that we will get only those test result data. We may get sometimes different also. For example, stability study. I think you know about stability study. Stability study information will be in a different aligned way than this chromatographic information. The chromatographic will be with a graph, all those things, but generally the stability study will be numerical numbers. So date, time, likewise. Okay, these kind of differential data also will be available in pharmaceutical sector. Those things can be used by laboratory information management system. Mean, please try to understand, we have three major system. One is graphic data management system, and then laboratory information management system, and then text information management system. So this chromatographic data system, we can use only chromatography and related informations. Okay, so that sequencing and then that flow will be totally different. Apart from chromatographic information, the industry will also have some other differential information. For example, we'll talk about stability information. This kind of number of differential information will the company will also have. So for to manage these kind of differential information, the companies will be having one more software is known as laboratory information management system. Clear? And then text information management system. This you can add, or you can upload any kind of text, image, photographs, these kind of informations, but generally they are not using nowadays. But on initial basis they were using. Okay, text information management system. Here you can keep photos, paragraph, anything, any information based on your wish. You can keep it, and then you can leave it. Okay, so try to understand. We have discussed about history. Okay, in history, how the computers were entering into pharma sector, and then we started discussing application of computer in pharmaceutical R&D. So first application we talked about, it is like a data analysis and data management tool. Computers are used as research data analysis and research data management tool. Next is computers are used in predicting metabolism. Okay, they are used to predicting metabolism. I think now you can give the hypothesis. Who will metabolize a drug? I don't want to go with this monotonous talk. Okay, anyone who can metabolize drug or in body, who will metabolize the drug?
Liver.
Liver
Enzymes.
is an organ. I'm talking who?
Enzymes.
Enzymes. Good. So enzymes are made up of.
Proteins.
Proteins.
Excellent. Proteins are made up of.
Amino
Amino
acids.
acids.
Amino acids.
Wonderful. Okay. So do amino acid have their chemical structure?
Yes, sir.
Yes, sir.
Excellent. So now you tell me, do enzyme have their own structure?
Yes, sir.
Yes, sir.
Okay. Okay. So once you know the structure of any chemical compound, be able to predict the molecule binding or not?
Yes, sir.
Yes,
Okay.
sir.
Once if you know it is binding or not, once after understanding the energy variation, we can also able to predict whether it can metabolized by this particular enzyme or not. That also we can able to predict. Are you getting what I'm telling?
Yes, sir.
Yes, sir.
Yes,
So,
sir.
well and good. So this is what now we are going to start talk closely about how in pharmaceutical area, specifically pharmaceutical area, these computers are utilized. So I think no, in protein data bank we do have enzyme structures, and then we have molecular structure. We know, if for example tomorrow some chemist is coming to you and then he's giving some new molecule in the sense. Okay, if you will ask, do you know the molecular structure? For sure he will be knowing the molecular structure. So you will draw the molecule and then you will drop into the software, and then this software will give the probability how much percentage it will bind to the enzyme, and then like what kind of reaction you may expect, or whether it is stable in this enzyme oxidation or reduction. Okay, like or glucuronide conjugation, these kind of enzymatic reaction, whether it is stable or not, those informations you can get it. So you think no, it will reduce tremendous work what to be done in wet lab. If not, in wet lab we need to those drug in each enzyme, and then you need to take it, quantify it, to do micro level assays, and then we will be finding whether what kind of metabolites came, and based on the metabolites we'll be telling whether the reaction happened or not. It is hectic procedure. We will do it, but we will not do for number of molecules. We will drop 100 molecules into this simulation, so it will tell three molecules are stable. Those three molecules we will check once again. Okay, and so we can reduce enormous amount of workload. So now we are talking more closely about how it is how those computers are exactly used in pharmaceutical related application. Okay, so the first application is in sense like predicting the drug metabolism. Okay, so this can be prediction of site of metabolism. So these are the things can be done by softwares. Prediction of site of metabolism. In enzymes also we do have a number of sites, and then each sites will be specifically bind with certain enzymes, certain drugs. That site can be found, and then elucidation of potential metabolites and their chemical structure after metabolism. What kind of metabolite can happen, what might be its predicted structure, whether the metabolite is active or inactive form, all those things. Now you see like how easily we can get the data. First we can get whether it will metabolize or not. Okay, next we will get who is metabolizing. Third, we'll get information after metabolizing, what kind of structure we can predict, do the structure is potent or not, that also we can able to get the information. And then prediction of direct and indirect effect of xenobiotics and metabolizing enzyme. Okay, next we can also predict once if this drug binding, do the enzymes will face any issue, or do the enzymes can have, do the enzymes will get repaired, or will the structure will be changing. Effect of xenobiotics mean effect of drug on enzyme also we can understand. Like ketoconazole is the one of the good example for one particular action. Can anyone able to tell? Please will it, once when you take ketoconazole, what will happen to the enzyme? Any idea? It's in biopharmaceutics metabolism chapter. No problem, leave it. Okay, so what happens now, we have one more mechanism, enzyme induction, enzyme inhibition. Okay, once you take certain drug, what it will happen is that will induce the enzyme, and so when you take the drug next time, it will metabolize easily, it will not produce the action. So if that kind of time, we will be increasing the dose. Okay, one more different mechanism, enzyme inhibition. Once you take the first dose, it will inhibit the enzyme. Second time if you give the dose, the inhibited enzyme will not help in metabolism. So what we will do, second time we will reduce the dose and give. Likewise, no, certain things are there. It is nothing but drug acting on enzyme. So this, that also we can able to predict whether it will it will change the nature of the enzyme or not. Okay, so anyway the outcome is like we can able to predict clearly how these xenobiotics or these drugs, okay, will affect the enzyme, and then what are the expected benefits. These things also we can get. Next important case, computer in toxicology and risk assessment. Okay, so what happens, once to find a toxicity study, we need to spend $1 million. You may think toxicity study is very easy. Yes, it is easy when you do for only one or two molecules. For example, you think a synthetic chemist is giving you 100 molecules and then he is asking, do toxicity study. Okay, it is not at all easy. You need to select number of groups of animals, or you need to select a human model or something like that. You need to give a drug, and then you need to check each and every organ, or else every cell, how it is behaved, what on acute administration what happens, on chronic administration what happens. Likewise, you give one day, what is the toxicity reaction happening. You give it for 10 days, what is the toxicity reaction is happening. You give it for 3 months, what is the toxicity reaction is happening. These kind of things, in a research wise quantifying, it is very tedious process, and we need to spend millions of dollars. Okay, one by, it is very very important. You remember, you please think, no, if scientist is developing a drug is like a kid. Okay, creating a kid. We have a kid. Okay, and then that kid is like at the stage of delivery, at the stage of some particular stage, we are coming to know some very bad information about that. Okay, it is like you have spent enormous effort, and then you have spent enormous money, and then you have created a drug molecule. Okay, after this 8 years or 10 years effort, you are coming to know, okay, sorry, this is highly toxic, we are not supposed to bring this to the market. Now you think about their mindset. Okay, they have spent enormous money in this. So because if it has fails in toxicity, 100% sure you cannot able to bring into market. That is for sure. Even if it is fails in efficacy, you can do something, and then you can able to try something like that. But if a drug fails in toxicity, that's all, the future of the drug is gone. Okay, it is very difficult. It will never ever be possible to bring into the market. So such important toxicity study. So this computer also supports in getting these kind of data. So okay, we have number of software like pass qsar, and then test, and then like topcat. Okay, and then toxtree. Like number of softwares are there. Okay, once you run in the software, that will predict and that will tell what are the possible toxicity we can expect, or then what might be the reason of the toxicity. I think you will know, I think clearly, even for the toxicity is because of the binding of drug with the protein. Yes or no? Any idea? How the toxicity also due to binding of drug and protein, accepting or not?
Yes, sir.
Okay, so the same way the computer can also simulate it, and then they can find whether it is producing any bad effect or not. This will reduce enormous money and then expenditure and then time of a researcher. Okay, this is next. And then now comes our extreme case relevant area, computer modeling drug dispositioning, ADME. Okay, absorption, distribution, metabolism, elimination, all those things we can do very easily by using computer model. Why? In the sense, I think you might have learned absorption biopharmaceutics. It may happen like transcellular, paracellular, or vesicular. Likewise, so there are a number of methods are there to observe. Each are again based on the chemical imbibes. Okay, so the computer can able to simulate it, and then again, even though I think my PG student were working on this concept, admit software they have used, and then from that we got a result. Okay, that simulation is exactly equivalent to that of the simulation what we did in laboratory condition. Okay, so what happens generally in the sense, these computers will have ability. Okay, those homologous models, no, because they have ability to check. Only you give molecular structure, and then you give like it's pKa, pH, like it's the molecular basic character you give, and then this will have ability to check whether it can able to pass through the absorption. If it is entering into the absorption, what might be, it might be like active absorption or passive absorption, transcellular absorption, paracellular absorption. These analysis the system can do. Clear, this analysis the system can do. So now you imagine what is the outcomes, in the sense, if I do the same experiment in animal model or cell lines, it will take enormous time, and then I want to test each and every parameters. But by using software, within like half a day I completed the total experiment. I checked nearly about 25 to 30 molecules. Okay, then I designed a dosage form. Now you see, like, and then and then metabolism automatically, as we discussed, absorption we can do with this modeling. And then metabolism, as we discussed, again the enzyme is a chemical model which can bind, and then those things can be quantified. And then elimination, and then like distribution, again with the protein binding model we can do it. Like so we have of models, with that we can able to predict whether ADM is happening or not. Okay, so two major method of how we can do, in the sense, quantitative approach and then qualitative approach. There are two major approaches are there, quantitative and qualitative. By these two approaches, we can able to predict whether the model is correct or not, or whether the ADM is adding to this particular molecule or not. These things we can able to detect. Okay. Then computer simulation in pharmacokinetic and pharmacodynamics. It is again one of the very famous area where nowadays a number of people are getting jobs, and then like very close to pharmaceutics. Okay, pharmaceutics in the sense I'm talking about pharmacokinetics. Clear? So I think no in silico we can do two methods. One, you collect the information, from an experimental model, and then you drop into this kind of pharmacokinetic software. Then the software will predict and it will give you like what is the half-life of the drug and what is the elimination half-life, and then what is the AUC, and then like Tmax, Cmax, all those data the computer will give you. Okay, so this again will reduce your manual burden of doing calculation by using graph sheets. Point one. And then you can get an exact information about the drug behavior in the plasma. That also will be it. This information is accepted by regulatory bodies. So now you see the regulatory bodies how they are believing computer. Okay, these kind of, so that's why it is used in pharmacokinetics and pharmacodynamic modeling. Like Win Online, we do had in our college lab. Okay, it is very famous software approved by regulatory body. So you can do kinetic, we can do n number of kinetic calculation, n number of modeling approaches there. And then non-MEM, it is again one of the very famous software. Non-MEM is for population pharmacokinetics. So like for example, you do clinical trial, and then you do pharmacokinetic study for 100 people, 200 people. Those information you can drop in non-MEM, and then this non-MEM will give whether the drug shows individual variability or the drug behave formally in all human model. See, the major problem what generally can happen or people face in the sense, you will give a drug to a person A, 500 mg, he will get good theoretical efficacy. The next person of same body weight or same physical nature, you give the 500 mg, it will not work. Okay, so this kind of error should not happen. So for that, we will be using population pharmacokinetic models. Population pharmacokinetic model in the sense, instead of giving to one person, we will give to 100%, 200%, and we'll check whether kinetic profile remains same or not. Those things can be done with non-MEM. Okay, it will simulate and it will tell you like the drug behaves in the same way in all population, or it is not behaving in the same way in all population. If it is behaving in same way to all population, we can close your eye and we can sell into the market, or we can to the next stage of clinical trial. If not, we need to rethink, because once after launching, one person may be getting benefited, one person may not get benefited, and then the third person may be affected, may receive side effect. Okay, these kind of issues can happen that we can able to avoid by using this non-MEM model. Okay, so this is again used in pharmacokinetics and pharmacodynamic study drug. See, is it clear till then what I discussed?
Yes, sir.
Yes, sir.
Okay, still
Yes,
I need
sir.
only half an hour. I think ethics already completed. Only we need to discuss about plagiarism, patent, and then computer utility in market. Okay, I need just a half an hour. I will complete it. So I will give two options. You can plan and you can tell me. One option, can we try to take time from now to off, like I think it is like 12:45 to 1:15 or 1:20. We'll take it as a single stretch. We'll complete, or after like 1 hour break from 1 to 1:30 we can take and we'll complete. Not 1 to 1:30, sir, 2 to 2:30. See, I'll give two options. First option, continuously I will take from now 12:45 to 1:15. I will try to complete one option. Second option, now we'll wind the class and then we'll take from 2 to 2:30. What will be the better option?
Sir, we'll continue, sir.
For everyone? Sorry,
Yes,
okay.
sir.
I thought,
Continue, sir.
okay, you will not sleep, no?
No,
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 sir.
no, sir.
No, sir.
Okay, then fine. Okay, I think like a code, and then see, this is again very interesting part. I will not bore. Just I will try to give you a short way. Whenever the computer comes to the game, okay, whenever the computer comes to the game, central storage system, networking, all those things comes to a game, automatically the data infringement can also happen. Okay, data infringement in the sense, see, you will write in a paper. Okay, you are writing a secret formula in a paper, and then you are sealing it, and then you are keeping in your table, and then you're locking it, and then you're keeping key with you. No one will allow into your office, and then no one will touch your table without your permission. You have such a security system. This is one possibility. Second possibility, you are creating all this confidential information in a computer where the internet is connected, you are storing in a cloud base, and then you're leaving it. You tell me which is more safe.
Keeping
Getting my
security.
question, no?
Second one.
Huh? So the
Second
manual
one, sir.
system is more safe or storing in a computer is more safe? Sorry, okay, you will tell. Okay,
Second,
now
sir.
I will ask the same question in a different way. Which you can easily breach, or in which system you can easily hack and get the information? Whether the manual system locking in, keeping in a table with a number of securities, or just storing in the cloud. By in which method you can easily breach or you can able to hack and get the information?
Manual
Sir,
method.
storing in cloud, sir.
Complete.
Like a manual, okay, I'm accepting, but it is not so easy. See, whenever the technology is increasing, the same way the breach or the counter technology also will increase. So see, yes, the information retrieval is very difficult in first method, but in second method, the information retrieval is very easy. But the same way, the risk of maintaining information also difficult. Okay, because the people can easily take it. No, people can take any good hacker. See, being a pharma scientist, we may not be knowing all those hacking, all those things. We know to follow the protocol and we will save in cloud or somewhere, something like that. But no, a good hacker can easily take and breach the information. Now I think the people are telling, uh, I think Russia is stealing information about the vaccine, and then China is stealing information about the vaccine. Likewise, no, people are talking like this. Now you see, they know very well to save it perfectly, but even though the stealing is happening, right, by technical way, that's what I'm telling you. That is why the government and regulatory body realized whenever we are going beyond technology, the same way the code of conduct and then no like ethics to maintain the information should be also done. Okay, about that was those chapters. I think Satya sir told those things were covered already. So let me go to the next two topics. Okay, I'll try to complete it fast. I think ethics and contact is done. So how about you people work from home, study from home, all those things? Now we are going to one of the very important topic which is needed for you. Okay, use of computer and the internet in scholarly information retrieval system. Okay, how we are going to use computers in scholarly information retrieval system, and then how we are going to train your system, or how this happens, how when you search certain things, when you search like erythroblastosis fetalis, you will get certain information, how this is happening, how the information is retrieved, and then what are the methods that are available to retrieve the information. About that we will be discussing now, just for a few minutes. Okay, so what is the function or what is the purpose of information retrieval system? It is used to recover the like community information, or like how I can tell in the sense, no, I think you are well versed about like Google, all those things. There are a number of community information speaks. Number of people are providing certain information, and then that load in the central cloud. And then from there, based on your search options, based on your information, what you're throwing into that particular server, it will filter and then it will give you the system. Actually, what is the use or what is the function? Okay, so with the help of those information retrieval system, we can able to we can able to retrieve the information based on our interest. We can able to get information based on our interest, and then we can able to take like kind of challenging information without spoiling its quality, and then we can take it at any point of the place where you work. Likewise, no, there are a number of applications are there about collecting the information. Okay, the major purpose of this information retrieval system is used to do research or understand the thing what is happening. But for that, you should know only one thing: the information should be available in the database. Okay, how it works? Information retrieval system is very simple. You are going to collect pre-existing information, okay, which is already available in the database, by giving a sequential code or sequential content. You are going to take the information. For example, in a database we have hundreds of information about vitamin A. You think vitamin A, B, C, D, about all vitamins. There are hundreds of informations are already available in our database. We have feeded those things. Now you want to collect about only the vitamin A information. Okay, once you type the vitamin A, the retrieval system will track and it will find only the vitamin A, and then it will show to you. Okay, so for that, the major two reasons: the code what you're giving, it should be related with the database which is available, pre-existing database. and then it is our responsibility to make our database more nourished with a lot of information. So this is what actually information retrieval system. Please try to understand artificial intelligence and the information retrieval systems are totally different. Information retrieval system in the sense you are providing, you are already nourishing your system with a number of informations, and then you are giving a question which is aligned with information, and then for that question information retrieval system will give an answer. So this answer can be used for your research purpose. This is known as information retrieval system. Okay, if you're not clear, again I'm repeating it, very simple. It is like retrieving the information, or you're creating you're not creating any information. So it's just in retrieving. Tomorrow you can retrieve, I can retrieve, anyone can retrieve it, you will get the same information. If I give same kind of question, I will get same information. That is an information retrieval system. That is what nowadays it is most commonly available, number one. Now recently people are working on artificial intelligence. Okay, it is not information retrieval system. Artificial intelligence in the sense it will think, it will create information based on available information, and then it will create its own information and it will give us. That is AI. Okay, how it works in artificial intelligence, you ask one question, it will give one answer. If I ask one question, based on my question, it will give slightly changed and it will give the answer. Both, see, information retrieval system and then artificial intelligence, how I can differentiate in the sense, like I think I need any one of your name, please. Anyone, please tell
Abhishek.
your name. Abhishek, well and good. Okay. So far, information now, I think no, information retrieval system is very simple. I will ask, what is your roll number?
The 2.
2. Okay. If I give, who is the, like in information retrieval system, like roll number 2, who is like roll number 2, if I give, or who is the name of, what is the name of the person who be in roll number 2? If I give these kind of questions, it will give only the answer Abhishek, Abhishek, Abhishek. Fine. So if I tell, if I click 2, if I enter, it will give Abhishek. If enter, who is the person who is occupying the space of roll number 2, it will give Abhishek. And then I will give, do Abhishek is really a roll number 2, it will give Abhishek. Likewise, no, if I give whatever the possible answer, it will give only these kind of information. It is loaded that 2 is related to Abhishek, so it will give this kind of information. Abhishek, Abhishek, Abhishek. This will give only this. This is known as information retrieval system. It is very good. Information retrieval system, you will get a pinpoint information, okay, about pre-existing data. But when it comes to artificial intelligence, if I tell, do Abhishek really a roll number 2? Then the artificial intelligence will tell, yes, actually the roll number 1 is this person, roll number 3 is this person. So alphabetically calculating, he is the 2, so you don't worry. This is artificial intelligence. Okay, the way how I ask the question, it will also give its input with the available data, but it will give in a, it will also think and it will give. Okay, those things, they're not generally promoted in pharmaceutical sector, just the research is happening. As of now, the information retrieval system is available. Okay, how information retrieval system will work? It will have three layers. Okay, the first layer, second layer, third layer. The first layer, the addition of records into the program and the development of the inverted documents. Mean, like in first layer, you can keep on add programs, and then you can keep on add information. The first layer is nothing but information. There you will be having lot of information. Second layer, this is here where the query process happens. Okay, so first layer which contains information, second layer here the query what is coming, no, that will process in the second layer. The third layer is nothing but user interface layer. User interface layer. Okay, so the query will come to the third layer, and then it will come to second layer, and then it will come to the first layer where the results will be given to the people. Okay, so what are the factors can affect information retrieval system? Please try to understand artificial intelligence. The affecting factors are very less because the system will think. Even if you did error, the system will correct. But information retrieval system, there is a chance of some error. So now we need to know what are the possible errors can happen in information retrieval system. So system factor in human factors. One problem, the computer is not good, the program is not good, or the retrieval process is not good. This is a system factor. Next is this human factor. See, for example, I'm asking you, please collect information about vitamin A. When you do a research, whenever you do a in MPharm project, what is the first stage? The first stage of MPharm project is learning to retrieve information from internet. That is the first stage of MPharm project. Okay. Once you learned it properly, you can survey throughout the world. Okay, but that is what people will never learn. The first stage of MPharm project in the sense very simple. You should learn how to collect information from the sources, internets. Okay, can be any source, but how to collect. For that, you should know what are the channels are available. Those things should be learned. That is the stage one. That's what I'm telling you. So what human errors can also happen. So you need to give a proper term. If you're not giving a proper term, you will get an irrelevant answer. Example I can give you: if you have a doubt, you go to the Google and search. You type tablet, what you can expect? You want to know about tableting technology, you go to Google and type tablet. Okay, and you tell me what age you got. If you have an access to computer, you can do it now. If not, no issues. Will you get a paracetamol or ciprofloxacin tablet, or you will get a no, digital tablet? You can check and you can tell me. This is the problem with information retrieval system.
Digital tablet,
Okay.
sir.
Digital
Digital
tablet.
tablet.
Huh? What you got? Tell me. You got digital tablet, right?
Yes, sir.
Yes, sir.
See, this is what information
Yes.
retrieval system. So what will happen? You know, this thing's happening from past 1 year. I'm keep on searching. Not even single. I'm not got no pharmaceutical tablet. I used to get only that mobile phone tablet. These kind of things. Okay, so then I used to think, array, people are so outdated or something like that. Okay, so let it be. So that's what I'm telling you. This is the problem with information retrieval system. Okay, so this is now you tell it is a machine error or your error, human error or machine error. You want to learn about pharmaceutical tablet, but you got this is whose error? Machine error or human error? What I can tell.
Machine, sir.
Hmm. So you're not ready to accept this is your error. See, you know very well we have two terms tablet. So this is not a machine error. This is human error. We should recoin our term word, and then we should give a proper question so that the information retrieval system will give proper answer. This is just an information retrieval system, so it will give these kind of answers only. But this is again, I will tell it is a human error. So we should give a proper question to the system. Okay, so these are the two errors, and then system factor is considered in term of the system efficiency will be less, and then the system interface will be less, and then sometime it may not be able to correctly understand what query you're giving. Okay, so there are number of errors which can happen in those information system. Generally human errors, what are the errors can happen? First, indexing error can happen, searching error, screening error. Like indexing error in the sense, when you try to give the information, that time itself you may be doing error, and then when you try to search information, that time you can do the error, and then you may get 10 informations. Out of 10 information, you will take we may be taking two substandard information. That is again a screening error. So the in information retrieval system, yes, the system can also do the error the same way. Equivalently, human, we can also do the error. When you go and search in Google, it will give you like 100 information, but apart from that, it is your responsibility to best of two. Google will already will give you best of 100 information, but it is your responsibility to take a good two information. So this if you have not taken selected properly or screened properly, we are not supposed to blame the information retrieval system. We can blame ourselves. Okay. I think these things I have covered. So what are the kinds of information retrieval system? We have two kinds: in-house information system and then online information retrieval system. This is very simple example. In-house information retrieval system in the sense, like we'll think about our JSS college. Okay, I will create a server. I'll create a server. So in JSS college Wi-Fi or in JSS college connecting devices, you can able to access the server, and then you can collect information about pharmaceutical preparations. Okay, just now, even next time and do that experiment, not a big deal. Okay, in a JSS college server, I will create I will create a cloud, and then I will create a server. In that, I will make it like only our under our college Wi-Fi, we can able to access the server, and then we can able to access the information. In that server, I will keep over 100 books. I will keep 100 books. Okay, so within our college premises, within our college Wi-Fi access, you can able to download it. Clear. And then if you have our college connectivity, you can download it. So this kind of is known as in-house retrieval system. Mean within your organization or within a controllable limit, you can able to put an information, and then you can take an information. So it will be completely under the control of your employees, and so the confidentiality is maintained. This is one way of information retrieval system method of using this. And then the second way is online information retrieval system. So this is again kind of there will be a hard drive memory which will be in somewhere, and then the information will be in hard drive in term of cloud, and then anyone can use the information at any point of the world, because it is forecasted or procasted in the online media. So this is again no online information retrieval system. Like I can give you an example of internet search engines is one of the best example of online retrieval information system. Anywhere you can retrieve the information, right? So likewise, no, in online retrieval information system, we have four types: OPAC, and then online database, and then digital library, and then web search engines. This more or less, this four are more or less same. Only the way or the only the content will be different. Okay, this OPAC you will get more kind of no written documents, and then like subjects information. These kind of informations will be getting, and then in online database you will get a literatures, these kind of information, and the digital library web search engines you will get whatever. You will get a video informations, you will get a picture information, you will, I think, I think you're well versed about search engines, right? All types of certain first two few information retrieval system, you will get only the written kind of information system. Rest two information systems, retrieval systems, you will get all informations, like starting from photos, videos, these kind of informations you will get very easily. Okay, so these are the four types of information retrieval system which is available in online mode: OPAC, online database, digital library, web search engine. Please try to understand. 2 minutes I will give from first till then a short recap. First we discussed about basic introduction. Next we discussed about history, how it happens in mid 90s to till end of 1990 we discussed. Okay, history, how the computer entered, all those things. Then we discussed about how the computers are used in pharmaceutical area. Okay, we started discussing about how it is used in data management, and then how it is used in toxicology, how it is used in like pharmacokinetics, and then how it is used in those like ADMA, all those things we discussed. And then we started discussing about like how it is used in information analysis, and then what is information retrieval system, and then what are there. We have two types: in-house and then online. In the online we have still four more types. Okay, and then what are the components, layer 1, layer 2, layer 3, what are the components are there in information retrieval system, and then what are the errors can happen in information retrieval system. The errors can be majorly two types: one is like machine error, and then the human error. The human error can be again three human errors can happen: indexing errors, screening errors, all those things. So like I think no, as of now we discussed these things. Is it clear? Because I'm just no, we need to complete it first, that's why I'm rushing it. As of now, you're getting any graph, no, what we are learning base at least to 10% or 20%? Are you really getting what we are learning actually?
Yes, sir.
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes, sir.
Okay,
Yes, sir.
thank you. Okay, now it is again very interesting part we are going to discuss: patent. Okay, so before entering into this information and then technology behind patent, we'll talk what do you know about patent, what is the use of patent. You give me 1 or 2 points, no, with that I can take forward. According to you, what do you know about patent, what is the actual use of patent, and then why this term is famous nowadays. You can give any information. I'm not asking answer, I'm just asking any basic information.
Sir, it gives protection to our innovation.
Exactly. So do technology can be patentable? Mean like software and then this computer based technology, any idea?
Yes, sir. Some
Yes.
softwares are being patented, sir.
Yes, okay. So initially they were not having this provision that to be done by only copyright or trademark. Now they have started giving these provisions to even softwares. Okay. So anyway, now we'll talk about patent. So when you developing, for example, you're doing a project or something like that, then you aim to know your project is novel. How you will come to know your project is novel? The first stage of patenting is you should know whether your product is novel or not. I'm preparing paracetamol tablet. Do you think it is novel? I'm preparing 500 mg paracetamol
No,
tablet.
sir.
Okay. If I seeing pharmacopoeia, do you think it is novel?
No, sir.
Can I able to file patent?
No, sir.
No,
Yes, we
sir.
can. We can file patent. You pay 1,500 rupees, anyone can file patent. But only problem, patenting will not happen. You will not get the
Full.
certification of patent. You can file, right? Anyone can write civil service exam, right? But becoming an IAS, only the person who selects in that exam will become an IAS. The same way, anyone can go for patenting, but the way completion of the all those criterias will not happen. It will be failed. Okay. But my question is very simple: what instinct will make you to think your product is patentable or not? What first instinct? For example, when you have a project in your mind, and then when you think your project is novel, right? What will make you to think your project is novel? My question is very simple. Anyway, you are going to do some projects, right? So what will make you to think your project is novel? That is my question. The question is very simple. You can try to guess any answer. Okay. Like, for example, you're coming and proposing to your guide about a project. Okay. So you will tell, sir, this is a new project, sir. No one has tried it. Okay. Something like that. So what will make you to think in that way? That is my question. What will give you confidence to tell to your guide, sir, this is novel or new? No one have did this. Something like that. What actually will make you? You think you will get an answer. Very simple way. Any guess? Check. I will level. Okay. We will search in Google. We will search in Google Scholar. We will search and we will read some papers. Okay. We will do in that. You will come to know this kind of research is not repeated anywhere. No one has worked with this technology, something like that. This will give you the confidence to go with your project. Yes or no?
Yes, sir.
This is what I'm expecting, but no one is telling. Well and good, no problem. Okay, no problem. No problem. Okay, so try to understand. So this is known as prior art. This is known as prior art search. Okay, whenever see, for example, no, the patent agent, what they will do in the sense when you come and then you tell, sir, I have some new project or something like that. The patent may not be necessarily, from your department or your domain or your qualifications. Okay, he may not aware of your technology, but a patent agent will know how to do the prior art search. That is the stage one of patent. Okay, so generally I'll tell you, frankly speaking, a patent agent will ask around 40,000 to 50,000 for filing a single patent. Okay, so what he will generally do in the sense, when you give your invention, when you give information about your invention, this person will ask you, you give first a small basic write-up. I don't want entire details about the patent. I want to know only a short abstract about the patent, and then you will give your abstract. This he will go for prior art search. This term is known as prior art search. Prior art search in the sense, before filing a patent or before publishing it as a patent, you are going for a basic search. So in this prior art search, there are number of websites are there, or else number of search engines, and then sci-finder, like number of technical tools are there, software tools are there. Once they will drop this patent into that software tools, and then they will start searching. So the software tool will run, and then it will give the information that whether this is patentable or not, or is there any research which copied already this and then have been exited or have been done earlier or not. This will tell. So based on that, it will tell clearly where are the places and which are the countries these kind of researches have been. It will search all throughout the globe. They will search all patent database, publication patent database. Okay, that in the search engine, it is again very costly, okay, to buy those search engines. But the search engines will search everywhere. It will search all patent resources throughout all countries. It will search all research publications throughout all countries. All those things it will search. At the end of the search, it will tell you it is 25 to 30% already pre-published in some other countries or something like that. So your agent will inform you, sir, it is something happened already like this. So we need to either redo the research by modifying these tests, or this cannot be patentable. Likewise, these advice you will get from your prior arts researcher. Okay, so what is the use of prior art in the sense, it is a patentable research mean, once if it is success in prior art, most case it is success in patent. Okay, but for to know the patent is succeeded, you need to wait for 1 year. But in prior art, within a day, you can able to understand whether your work is novel or not. So patentable research we can do, and then patent examination we can search. Examination search also we can understand, and validity search we can understand, and then state of art, like other patents validity you can understand, other patents whether it is what kind of the other patents are novel from your patent, everything. A to Z of patents you can able to search from this prior art search engines and prior art softwares, those database understanding softwares. Okay, so what is the outcomes? Application, it will enhance the patentability of your work. Okay, so by seeing the prior art, we will get a clue. I will tell you, without doing prior art, if you do direct filing, what happens in the sense, it will go for publishing all things. Finally, the patent attorney, the person who is going to give the patent, he will ask you, this is already published in Europe 1945, you have not checked what to be. Likewise, so that time you will be stuck up, you may not have any answer, so your patent will be rejected. So these things can be avoided by using prior art search. So then it can minimize the total cost. Okay, why this prior art search can minimize your total cost? In the sense, this prior art is very cheap, like because like it will cost around 5,000 or 4,000 rupees. Once you do the prior art search, what can happen, you will understand whether we need to go for or not. See, we may be just searching in Google Scholar or Google or PubMed, all those things, and then according to you, you may be thinking the product is novel. But once you go for prior art search, it will tell 60% is not novel. Already the contents are similar in the society. Okay, and then you will get an idea whether I need to go forward or not. Without knowing this, if you go forward and then you spend a lot of money giving money to agents and then agencies, all those things, and then at the end of the day, if the patent got failed, then who is responsible? Right? No one. So that's what no, they'll minimize most of the companies, most not I'll tell 100% companies. We will do even in JSS, we will do first a prior art search first. If a student come with a novelty, if even do I got some novel idea, what I will do, I will go for prior art search. It's one of the very good tool, okay, which will reduce our work burden and then it will give you confidence whether the product is patentable or not. Okay, I will do prior art search and then I will from that I will understand whether my work is patentable or my student work is patentable or not. So I will reduce the money burden and then I will reduce the effort. Okay, like for example, making everything completely, then at the end of the day, once after going to government agency and then they are telling it is not patentable, we are wasting our effort, right? Before that I will go for prior art and then I will to make it novel, or I will do some adjustment in the experiment to make it novel, or sometime if it is 90% copied, then I will reject the work, I will not take it forward. Okay, so that's what no, it is again no, that is the next important of prior art. May implies where the profit should be allotted. Okay, for research development, if any undiscoverable field is identified, this is again prior art search very important in term of pharmacy area. Okay, so once if, for example, you four members are coming with some novel idea, I'm searching in prior art, in that I came to know one work is completely novel, no one have touched that area. So now what I will do, I will allocate more money in that area and then I will try to work and then I will try to create patent, create a product in that area. That also we can able to find. Okay, may provide a review on like infringements, or like how I can know, I'll come to know like is there any infringement in the sense no any deviation in the law, any deviations, or anyone is trying to break our patent, or anyone is trying to copy our work, all those things can be done. Okay, then investigator legal document, these things can be searched like question. See, that's what I'm telling you. Once you go for prior art search, you can get all information related to the existing patent, and then all informations about our patent, whether it can be patentable, our product can be patentable or not, and then information about like how to be in, how to carry forward those patents, all those things. Okay, now to search a patent, in that when time permits, you please read these points. Okay, so generally, I think you know the term plagiarism. I think, okay, I'm not sure, but do you know what I mean by plagiarism or plagiarism, something like that? If you know, you can tell, no issues.
After completion of our project, we just check with the plagiarism. Whether,
That is good, very good, very good. Whether
whether
it is
the content is taken from the any articles, how much we have taken from the articles and copied from them. It is going to check it, sir.
nice, very good, very good. Okay, so it is very simple. Plagiarism will give you an idea whether these words or whether these concepts are copied or not. Okay, so now my question, like a water heater is a term. Okay, we can also use the term geyser. Okay, solar water heater is a term, we can also use solar geyser. These two terms gives the same meaning. Cell phone is a term, mobile phone is again a term which gives the same meaning. Communicating device is again one more term which gives the same meaning. Okay, now we'll talk like this. A smart scientist got a patent on mobile phone before 3 years. Okay, but this person in his total patent manuscript, he have never ever used the term mobile phone in any of this. It is like 10-page patent, you think 10-page patent file. In this 10-page patent file, he have never ever used the term mobile phone. He have used only communication device. Likewise, he developed a technology. We'll talk now some example. We'll talk. He developed a technology. Once you think the number, yeah, the number will be dialed. Once you think call dad, if you think, then the call will go. Likewise, he developed a technology. He patronized the technology, but unfortunately throughout his manuscript or throughout his patent file, he written communication device, communication. He never ever used mobile or cell phone throughout the script. After 5 years, after 2 years, you also thought of the same technology. Now you are trying for prior art search, but unfortunately, without using the term communication device, you are searching cell phone, mobile phone, something like that. So at the end of the prior art search, it is telling your product is novel. So what we will do with full confidence, we'll go for filing, we'll wait for 1 year or 2 years when your product, and then when it comes to inquiry, you will come to know the product is already exist. So now tell, it is fault from your side or not? You're
Yes,
understanding on the case?
yes, sir.
Are you, so it is fall from our side. So try to understand. So that is what, whenever you go for a search, the search should be with multiple, you need to use multiple words and then multiple terms. Likewise, you need to give complex terms, and then you need to keep on search with multiple words and phrases, something like that. So then you will get a clear cut information. That is how the information retrieval system works. Okay, so you will get a clear cut information whether this is exist or not. For example, you use the term a novel cell phone, again you can use mobile phone, again you can use only mobile, and then you can use communication device, voice communication device. Likewise, you can use number of combinative words. So you will come to know, you can able to collect all information related to these terms, and then you will come to a conclusion whether it is repeated research or not. So when it comes to a tablet, also the same, where you need to use multiple combination names and then multi chemical names and then molecule names, all those things. Okay, when drugs and dosage form. Okay, likewise, you need to give multiple terms so that no, the information retrieval system or pattern search engine or a prior art search engine will try to collect all the information related to the term. If you left a term, then the prior art search engine will remove it and then it will collect other terms. Sometime the old researcher or someone know who may use that term which you might have missed to listen. So that's what it is very, very important. And then the such procedure also given clearly, you can see when time permits. Okay, so how to search all those things, this is a patent, all those things we discussed. Next, plagiarism. Plagiarism, when you do a research, the one of the major okay, so again, it is very, very important to know what is plagiarism and then how to do the plagiarism analysis. Okay, so these things I will just try to give you a short capsule content. Then if you're still, if you want to know about those things, you can ask me, I will teach separately again. Okay, so try to understand, whenever you do a research, the very important thing is we should know how to, I think there are a number of softwares are there, Turnitin softwares are there, and then plagiarism analyzer, and then CO2, like this number of softwares are there. Okay, whenever we do a research, it is very important to understand whether the research is done by other or not, or the research the research is published by other or not. See, the research and then science is always like a marathon. Anyone can do a research, anyone can do a research, but the person who is communicating the first, he will get the credit. Very simple, this you should know. Okay, people are, I think, you know about the moon, and that I think the person is Neil Armstrong. No one is talking about who designed the rocket, who designed the space rocket, and then who sent the space shuttle. No one is talking about that. The person who is kept the feet on the moon, the people knows the name. Okay, we don't even know who is the senior scientist of the space shuttle designer. Okay, we are not ready to talk about him. We are ready to talk about only the person who is kept feet on the moon, because he is the person who communicated properly. That is what I'm telling you. When even in a research, you might have started research before 10 years. Okay, but you have not communicated any research papers, or you have not communicated it to the society, either in term of patent or publication or something like that. Society will not talk about you. A person might have did the same research before 1 year. He started communicating to the society in term of conferences, in term of publications, in term of patent. People will remember him as the scientist, not you. That is what, no, very, very important thing. So that is how the scientific community introduce the term plagiarism. Plagiarism is very simple. It is, we will tell, text to text plagiarism, pre-processing plagiarism, and then like a diagrammatic plagiarism. Likewise, number of types of there. A number of types are there. Once a paragraph written by you, okay, the plagiarism may be two-way. One is like verbal plagiarism, another is technology plagiarism. Mean, you can write the same technology, it is again a plagiarism, and then you can use the same word, that is again one more plagiarism. So when you do a research, we need to avoid both. Okay, we should, we can, you're not supposed to We can follow the technology as such. Okay, preparing nanoparticle, you follow the technology, but you will not get the patent. Okay, because the technology is already plagiarized. Okay. Second, you may use the same word as such, that will reflect in plagiarism. If you use the technology by using the words, for example, take 1 gram of sample, take 1 gram of sample, mix in hot water. This is the technology. Take 1 gram of salt, mix in hot water. This is the technology. Procedure is the technology I'm talking. Okay. You are take 1 gram of salt, mix in a hot water, mean you're using the same words. This is said to be as a plagiarism, which should never ever happen in pharmaceutical area. Using the same technology, using the same word is kind of a copycat. So that we have number of softwares, Turnitin, likewise, softwares are there, which will never allow these things to happen, which will find this easily. Okay, which will find this easily. So that is known as plagiarism. Then technology repetition can be found by prior art. Technology repetition can be found by prior art. Mean you are preparing a nanotechnology by using co-preservation phase separation method. Okay, you're using preparing a nanoparticle by using co-preservation phase separation method. This is a technology. Once you do the same technology by using words in a different way, if you're using the word, this will not reflect in plagiarism, but it will be get caught in prior art. So that's what I'm telling you. You should know to use these softwares perfectly before entering into the research. Once you know to do prior art search, once you know to do plagiarism check, all those things, once you know, then you will understand how the science community is tracked, and then know from that you can able to think in a different research. Before, if you have a plan, you go for plagiarism check and then you go for the prior art search, then you will know your idea is novel or not. Once your idea is novel, in the sprouting stage itself, you can able to change the idea. Okay, generally no, our students, what they will do in the sense, after making the idea into a product, they will go for this kind of search, which will not work out. Okay, then they will be wasting their time. So whenever you have an idea, you need to go for these kind of searches. Then you will come to know whether your idea is really novel or not. Okay, we have number of tools are there, like Dockle and then Urkund, all those things. MOS tool is there, very famous, and then JPleg are there. We are using Turnitin, which is highly we are using, I think we are using this Turnitin tool. Okay, this is very sophisticated tool. From that you can able to collect very whether it is plagiarist or not, you will get pinpoint, you will get. Okay, like you need to collect a document, and then you need to go for pre-processing, and then it will go for text classification, that will classify into text into two types, like information extraction, and then it will summarize the information, and then finally it will process the information, and then again it will go for text classification. Likewise, it will go multiple layers of text classification. It will break, for example, if you give a paragraph, it will break the paragraph into lines, it will break lines into words, and then they will tell whether the word is repeated in any research or not. Likewise, no, it will break into pieces, multiple layers of text classification, and then it will tell whether it is plagiarized or not. Okay, if it is plagiarized, then you did as such copycat, which is not considered as a ethical research. Okay, so this is what happens. Next, it is like market analysis. So computers in market analysis, in the sense, like what are the ways the computer used. This is not closely related to the pharmaceutical sector. Okay, but generally, no, it was used for forecasting the market. Forecasting the market in the sense, like if that we cannot know, we may not be knowing, some supercomputer might have already forecasted the COVID will come, we need to develop vaccine. This might have happened. Okay, in some big companies, we may not be knowing that, but this is what market forecasting. Okay, so you will give what is the market need, and then what is happening in the market currently to the, you will feed everything to the computer, then computer will give you the idea. Okay, what it will give you in the sense, like these are the changes happening, so you do research on this area, or you try to push the market product to this particular region, something like that. No, the computer will give you the input, then accordingly the company will start marketing. Okay, like it is used for improving market precision, like how much to be sold, and then what is selling, what is not selling, all those market precision we can get it from computer. And then like it will increase your camping capacity in the sense, like once you use internet as a platform, you can sell anything. Okay, I think you can check Instagram, you can see number of ads coming there. Okay, so that is what, no, in local market, when you do channels, Tamil channel or something like that, it will be in only in Tamil Nadu. If you do English, it will be in, it is again a regional marketing. But when you go for internet, it is throughout the globe, so again it will increase the capacity of market camping. Mean throughout the world will use to check Instagram, Facebook, all those things, right? the market capacity is increasing, number two. And then it is automate market camping. You don't want to push button every time to market all this to make it like that. You feed it once, then automatically whenever you start searching about that particular product, the advertisements will come, and then the market will increase. Okay, so that is what, no, you go to Google and search something, slipper or cosmetic, something like that. When next time when you go to Facebook, or next time when you go to Instagram, you can see the similar advertisement is coming. This is automatically generated because it is pre-programmed. So this is again no automated marketing. It will effortlessly, the product will be marketed automatically. People start buying it. Okay. Then it is again provide effective, efficient sales support. Like you go online purchase, it is completely efficient. In Amazon company, no morely like 2,000, 3,000 people might be working. Reason, it is very simple. Like you do online purchase, the advertisement is online, the purchase is online, the cash credential, everything is online. The computer is facilitating all those things. Improve collaboration, and then reduces cost and time spent on market analysis. Once we know everything, we know, for example, you see this particular cosmetics in which region of people is searching more. Okay, this is again automatically analyzed. Okay, you will get the data which is automatically generated. This is known as ROI, will tell, return on investment. Mean, like you need to invest a lot on softwares and then all those programs, but the return is very good. Okay, because you don't, without effort or reduced effort, you will get a very clear picture on which place sales is not happening, which place sales is happening, and then where to be more marketed, and then automatically marketing advertisements will be done automatically, and then the purchase also, know the money transaction, and then sending a product, everything will be done automatically with the help of computer. So this is actually computers in market analysis is very widely used. Number of visualization effects, and then like graphical effects, and then like um, A to Z, the computers are highly used in market analysis. You can see like visualization, real-time customer engagement, and then like I think you can see you will get you can get SMS advertisements, you will get an email advertisement, and then you can see when you go to websites, you can see, inside it will ask chat. Once you type hi, the computer will give you the answer. Okay, likewise, there are number of advanced settings are there. That's on a personalized marketing and consumer engagement. What you need, it will be personalized, it will be chatting with you, and it will engage with you, and then it will resolve your queries automated way, and enhanced social media strategies. Okay, like I think in Instagram, Facebooks are now the very big media for marketing. Okay, it is very cost effective, and in the same way the reach is very high. And then real-time customer engagement. If you have a query now, it will give you the mail, and then the computer will start trying troubleshooting the queries automatically. That is again real time. And then visualizing data, it will give more graphical information so that you will understand easily. Okay, then customers behavior, it will try to understand the customer's behavior, and it will try to show the product accordingly. If you have a doubt, you can just search in Google about some cosmetics or some slipper or something like that. Then you go to Facebook or Instagram, it will give advertisement accordingly. Likewise, it will know what you need, and then it will try to show the product accordingly. Everything is automated. So those strategies, everything can be done by computers. I think here I have given certain softwares which is used for market analysis. Okay, so in this, the Tableau is very famous software. People who is very strong in Tableau will get salary in lakhs. Okay, so I think these are the questions. We have covered this subject. Yeah, so any doubts you can ask me. I'm just, let me stop sharing. Okay, I think it is very long class, I'm sorry. I think okay, so it's completed. Any doubts, just you can WhatsApp me or you can mail me. So I'll try to clear your doubts. Okay, now you can go for lunch, something like that. Okay, so thank you.
Thank you, sir.
Thank
Thank
you,
you.
sir.
Thank you, sir.
Thank you.
Thank you, sir.